Hello, everyone. Welcome to another session in neurophysiology. Today, we shall see the role of the cerebellum in the control of body movements. So we shall see how the cerebellum performs these functions of controlling voluntary and coordinating voluntary movement in the body. So this is the outline. We shall see the roles of the cerebellum in the control of fine movement of body extremities, in the planning and time sequencing of movement. We shall see the role of the cerebellum in the maintenance of balance and equilibrium during movement, and we shall also see the control of ballistic movements by the cerebellum, and we shall see the mechanisms that are used by the cerebellum in damping and facilitating of movement. So we shall start with the functional anatomy before we move to the functions of the cerebellum when it comes to control of body movement. So we move to the cerebellum. The cerebellum being part of the metencephalon. Remember, the metencephalon is made up of the pons and the cerebellum. You can see the cerebellum here situated posteriorly to the brainstem. So anteriorly to the cerebellum, you can see the pons. So the pons and the cerebellum are parts of the metencephalon. So the cerebellum contains about 50 billion neurons and is the second largest structure of the brain. Unlike other parts of the brain, the distribution of the gray and the white matter in the cerebrum is the same like in the cerebellum. So remember, the cerebrum contains the outer gray matter and the inner white matter. So the same distribution is seen in the cerebellum. So we run to the parts of the cerebellum very briefly. So briefly, we shall see the vermis, which is the narrow portion of the cerebellum. Then we shall see the cerebellar hemispheres. This is a superior view of the cerebellum, and this is a mid-sagittal view of the cerebellum. So you can see the lobes of the cerebellum, the left and the right lobes. In between is the vermis of the cerebellum. Then the vermis has 9 parts, is divided into 9 parts anatomically. We shall not go into details of the division of the vermis. So part of the vermis on the upper surface of the cerebellum is known as the superior vermis, and the lower part of the vermis is known as the lower vermis or the inferior vermis. So you can see the 9 parts of the vermis being numbered here. So the vermis, like I said, is divided into 9 parts. So these are the names of all the 9 parts of the vermis. You can see the numbering there. Then, apart from the vermis, we move down to another structure of the cerebellum known as the nodulus. The nodulus is continued on either side. When you move down posteriorly or inferiorly from the vermis, you reach the nodulus. The nodulus has continuations known as the flocculus. So the nodulus and the flocculus are together known as the flocculonodular lobe. Anatomically, we have divisions of the cerebellum as anterior, posterior, and the flocculonodular lobes. These lobes are divided primarily by fissures. So you can see the coronal section of the cerebellum, or the mid-sagittal section of the cerebellum here on the left side of your screen. You can see the primary fissure dividing the anterior lobe from the posterior lobe. And you can see the posterolateral fissures dividing the flocculonodular lobe from the posterior lobe, respectively. So phylogenetically, when we talk about phylogenetically, we mean relating to evolution, evolutionary development. The anterior lobe of the cerebellum is part of the paleocerebellum, the anterior lobe there, phylogenetically. 90% of the posterior lobe here, 90% of this posterior lobe is recently developed. It's known as the neocerebellum. Then the flocculonodular lobe that we already seen is phylogenetically the oldest part of the cerebellum, known as the archicerebellum. Then functionally, we have three divisions of the cerebellum. Functionally, there are three divisions of the cerebellum. We have the vermis and the adjacent portions, known as the spinocerebellum. You can see the spinocerebellum here, and then you can see the functions it plays during control of motor movements. So we have connections with the descending systems, which are the corticospinal tracts, the anterior and lateral corticospinal tracts, as well as the extrapyramidal tracts. So we also have, still on the functional divisions of the cerebellum, we have the nodulus. Remember, the nodulus flanking and the flanking flocculus, they are collectively known as the vestibulocerebellum. The vestibulocerebellum. So the vestibulocerebellum has connections to the vestibular nuclei. The vestibular nuclei in the medulla oblongata. So it helps in balancing eye movements, the vestibulocerebellum. So we move to the next part of the functional divisions. We have the lateral portions of the cerebellar hemispheres is also a functional part of the cerebellum. So these hemispheres are known as cerebrocerebellum, the lateral portion. So you can see the connections of the lateral portions to the motor and premotor area. It has connections with the motor and the premotor cortices. It is involved in motor planning. So you take note of the functional divisions of the cerebellum and their role in motor coordination. So from the spinocerebellum to the vestibulocerebellum to the cerebrocerebellum. So these are summary of the functions of the different parts of the cerebellum that we just mentioned. The summary is on the diagram on the left side of your screen, and I also added more explanation to those functions of the different parts of the cerebellum. So that takes us to the functional anatomy. The cerebellum is made up of the outer gray matter, and which is the cerebellar cortex, and the inner white matter, like we discussed earlier in the first slide. So we said the distribution of the white and gray matter is the same as we have in the cerebral cortex. So the white matter, the inner white matter, is formed by afferent and efferent fibers entering and exiting the cerebellum, carrying sensory and motor information to the cerebellum. So we move to the next slide. The gray masses are cerebellar nuclei. The gray masses located in the white matter. There are some gray masses located in the white matter, known as the cerebellar nuclei or the deep cerebellar nuclei. Example of a deep cerebellar nuclei is the dentate globus, emboliform, and fastigial nucleus. So in the cerebellar cortex, we have cells and we have layers. There are mainly three layers: the molecular, Purkinje layer, and the granular layer. You can see the layers shown in the diagram here. You can see the molecular layer, you can see the Purkinje cell layer, you can see the granule layer, and you can see the white matter here. So we have cells as well in the cerebellar cortex. There are 5 types of cells or neurons from the Purkinje cells. As you can see in the diagram on the left side of your screen here, you can see the Purkinje cells. You can see the granule cells. You can see the basket cells. And you can also see the stellate cells. You can see even the Golgi cells. Those are the cells that are present in the cerebellar cortex. So you can note the layers of the cerebellar cortex and the cells that are present in the cerebellar cortex. So the main inputs to the cerebellar cortex is via the climbing fibers. The climbing fibers here are the main inputs to the cerebellar cortex, and also the mossy fibers into the cerebellar cortex. Those are the 2 main inputs to the cerebellar cortex. So you also have the main output from the cerebellar cortex is from the Purkinje cell. The Purkinje cell makes synapses with the cerebellar nuclei, while the mossy and climbing fibers also send collaterals to the cerebellar nuclei as they move into the deeper cortex of the cerebellum to make synapses with other types of cortical cells or neurons in the cerebellar cortex. So we move ahead. So the Purkinje cells, like I told you, the Purkinje cells are the main output, the final output from the cerebellar cortex. They have connections with the deep cerebellar nuclei. You see the yellow ones here are the Purkinje cells, which has connections with the deep cerebellar nuclei. So they have extensive networks of dendrites, and they receive excitatory signals from the climbing fibers. So as the climbing fibers and the mossy fibers brings input to the cerebellar cortex, they also make excitatory connections with the Purkinje fibers. Remember, the Purkinje are the main output from the cerebellar cortex. So that connection between the climbing fibers and the granular cells within some granular cells also make connections with the Purkinje cells. You can see the granule cells with their long parallel fibers, axons, connecting with the Purkinje cells. So all those connections are excitatory, and they have GABA as the neurotransmitter. But the Purkinje cells receive inhibitory signals from basket cells. You can see the basket cells here. You can see the connection between the basket cells and the Purkinje cells. It is inhibitory. So axons of the Purkinje cells form the only output from the cerebellar cortex and releases GABA as the neurotransmitter. So you can see the output here via the deep cerebellar nuclei, which now exits the cerebellar cortex to the descending tracts or up to the motor cortex. When we go to the granule cells, we are still on the excitatory cells of the cerebellar cortex. The granule cells receive excitatory input from the mossy fibers. The granule cells, they receive excitatory input from the muscle fibers. You can see the granule cells here. The muscle fibers, remember, they are one of the main excitatory inputs to the cerebellar cortex. So once the synapse with the granule cells, you can see the granule cells has long axons, which also communicates with other neurons. So the synapse with the granule cells, and then the granule cells sends signals to Purkinje cells via the T axons, the long parallel fibers, and it also sends to the Golgi cells, and it also sends to the basket cells here. So the connections between Purkinje cells, Golgi cells, basket cells, and the granule cells, the neurotransmitter in that connection is glutamate. So we are still trying to see the circuitry, the internal connections within the cerebellar cortex. So the granule cells innervate the Purkinje cells. The granule cells innervate the Purkinje cells. It sends axon to the molecular layer, like you have seen here. It has axons extending to the molecular layer, where the axons bifurcate to form T-shaped branches known as parallel fibers. Then we move to the other types of cells. We have seen the granule cells, the Purkinje cells. Their connections are excitatory. Then we move to the other types of cells in the cerebellar cortex. We have the Golgi cells, the basket cells, and the stellate cells. So those are the another three types of neurons in the cerebellar cortex, and they are also inhibitory in nature. They form connections with other types of cerebellar neurons. So the Golgi cells are located in the granular layer. You can see the Golgi cells here, located in the granular layer. So they have dendrites projecting into the molecular layer. You can see them projecting into the molecular layer, and they receive inhibitory information from the granule cells. The granule cells here stimulates the Golgi cells with an inhibitory information via the parallel fibers. Remember, the granule cells possesses parallel fibers that extends into the molecular layers and synapses with the Golgi cells. We also have the basket cells. The basket cells here are located in the molecular layer as well. They are stimulated by the parallel fibers of the granule cells. Remember, the granule cells has the long fibers that has connection with nearly almost all the neurons in the cerebellar cortex. So the basket cell is inhibited by the Purkinje cells. The connection between the basket cells and the Purkinje cells is inhibitory. You can see the connection here between the basket cells and the Purkinje cells is an inhibitory connection, and the neurotransmitter there between them, the Purkinje cell and the basket cell, is GABA. We also have the stellate cells, which are located mainly in the molecular layer here. You can see the stellate cells. They are similar to the basket cells, but are located in a more superficial molecular layer. So the connections between all these cells that we have seen, between the Purkinje cells, between the basket cells, the stellate cells, the granule cells, that connection between all of them in the cerebellar cortex is known as intrinsic cerebellar circuitry. Intrinsic cerebellar circuitry. That's the name of that connection there. So as a result of muscle and climbing fibers being the excitatory inputs, the main excitatory inputs, you can see muscle fibers coming in there. We have muscle fibers bringing impulses from proprioceptors, from the vestibular nuclei, from the radinucleus, and we have climbing fibers coming from the inferior olivary nucleus, the inferior olivary nucleus in the medulla. The muscle fibers brings impulses via the spinocerebellar tract. Remember, the spinocerebellar tract is one of the ascending sensory tracts. So as a result of these two excitatory inputs to the cerebellar cortex via the muscle fibers and climbing fibers, there is a kind of intrinsic cerebellar activity that occurs within other neurons, between the basket cells, between the granule cells, the Purkinje cells, the Golgi cells. So that connection that we had seen is known as intrinsic cerebellar circuitry. So in the intrinsic cerebellar circuitry, we have what we call a feed-forward inhibition. A feed-forward inhibition is a condition in which the interneuron, we have three neurons in the connection. The interneuron in the middle inhibits the neuron in front of it, for example. So the neuron that is inhibiting another neuron in front of it is receiving a positive or excitatory input from another neuron. So that is known as a feed-forward inhibition. So for example, we have a feed-forward inhibition of Purkinje cells. Remember, the granule cells are excitatory. So we have the granule cells feeding to the Purkinje cells, okay? So with the long T axons, we have the granule cells here feeding into the Purkinje cell. It's a positive connection there. Then we also have the granule cells feeding to the basket cells. The granule cells has connections with the basket cells, which is a positive connection. You see now. So the granule cell has a positive connection with the basket cell, while the basket cell exhibits a negative connection with the Purkinje cell. So that is how a feed-forward inhibition occurs within the cerebellar circuitry. So we also have, there are three types of them. We have seen the first one. We also have another feed-forward inhibition of granule cells, into the granule cells connecting with the Golgi cells, and the Golgi cells feeding negative connections to the Purkinje cells. So we also have the third intrinsic cerebellar circuitry, where we have the granule cell connecting with the Golgi cell feeding negative impulses or inhibitory impulses back to the granule cell. So that is an example of the feed-forward inhibition, how it works in the cerebellum. So you can see the connections. For example, interneuron A, C, and B. Interneuron C has a negative or inhibitory excitation. So it negatively inhibits interneuron B while it receives a stimulating feedback from interneuron A. So, for example, interneurons C can be basket and stellate cells in the cerebellar cortex. So they exhibit a kind of feed-forward inhibition to other interneurons. So that is how the feed-forward inhibition works. While the interneuron C in the middle here receives excitatory input from interneuron A, it sends inhibitory feed-forward to interneuron B. So, excuse me for that. So we have the feeding forward of negative impulses to interneuron B, while the C receives excitatory impulses from interneuron A. So what is the role of this feed-forward inhibition? What is the role? What is its importance in the cerebellum? So as we have seen, those feed-forward examples between the basket and stellate cells, between the Golgi cells and the Purkinje cells, That feed-forward inhibition is there to help in editing of motor plan by predicting sensory consequences, by correcting errors in execution of motor plans, and by smooth execution of the motor plan. So in other words, the cerebellum learns to associate motor commands with sensory novel consequences. So in the process of executing a motor command or a motor plan that has been prepared by the cerebellum and the cerebral cortex, the cerebral cortex and the cerebellum work hand in hand in executing a motor plan. So in the process of executing that plan, the cerebellum constantly receives sensory feedbacks from the proprioceptors to see how the motor plan is being carried out. It also receives those sensory impulses to see if the motor plan that has been carried out is the same as the plan that is stored within the circuitry of the cerebellum. So that motor plan is there. That feed-forward inhibition is there to help to maintain and match the motor plan and the sensory information. So if the sensory information that is coming in to the cerebellum does not match the motor plan that is being executed, then the feed-forward inhibition comes into play to correct. That is when error correction comes in. So the feed-forward inhibition will now come in to correct that motor plan to match with the existing memory of the motor plan in the cerebellum. So that is the summary of the function of that feed-forward inhibition. So we also have in the gray matter, remember, we have inputs coming in to the cerebellar cortex. So in the cerebellar cortex, we have two main inputs to the cerebellar cortex via the climbing fibers and the mossy fibers. I've told you where the climbing and mossy fibers come in from. So both mossy and climbing fibers are excitatory. The climbing fibers come from the inferior olivary nucleus in the medulla, and they project to the dendrites of the Purkinje cells. Before they move to the Purkinje cells to make synapses, they have already sent collaterals to the deep cerebellar nuclei. So that inferior olivary nucleus coordinates information from the spinal cord. It receives impulses via the spinocerebellar tract to the cerebellum to help in regulating motor coordination and motor learning. The climbing fibers excite the Purkinje cells directly and the deep cerebellar nuclei via collaterals. I've said it already. So the connections between these two structures and the climbing fibers releases excitatory neurotransmitter aspartate between the climbing fibers and the two other structures, the Purkinje cells and the cerebellar nuclei. So that diagram shows you how the circuitry of the cerebellar cortex works. You can see the climbing fibers as it enters, it has connections with the Purkinje cells here. And it also sends collateral to the cerebellar cortex. It's receiving impulses from the inferior olivary nucleus in the medulla as a result of termination of second-order neurons from the spinothalamic tract. We go to the mossy fibers. The mossy fibers provides direct proprioceptive inputs also from all other parts of the body, plus inputs from the cerebral cortex. They come into the cerebral cortex, into the cerebellar cortex via the mossy fibers. So inputs coming in from the cerebral cortex, inputs coming in from the proprioceptive organs, they enter the cerebellar cortex via the pontine nuclei. So that is the route of the muscle fibers. Remember the type of inputs that they carry. So the muscle fibers, after reaching the granular layer, as they ascend up into the cerebellar cortex, in reaching the granular layer, they branch profusely. They have many branches, and then each branch terminates within a glomerulus. Each branch terminates within a glomerulus. You can see an illustration of the glomerulus here. So you can see the muscle fibers coming in and forming a glomerulus. Okay, that's a kind of a glomerulus where its synapses, it will make multiple connections with different cerebellar cells, the cortical neurons there. So let's move ahead. So after reaching the granular layer of the cerebellar cortex, the muscle fibers branch profusely, and then each branch terminates with a glomerulus. That glomerulus receives axons of Golgi cells, as we have just seen in the illustration. So they excite the Purkinje cells through the granular cells. So once they have a connection with the granular cells indirectly, the granular cells can now in turn excite the Purkinje cells. Remember, the Purkinje cells are the main output from the cerebellar cortex via the deep cerebellar nuclei. So that is the connection there between the muscle fibers, the Golgi cells, even the parallel fibers of the granule cells and the Purkinje cells. So you can see how the connection goes. It goes within the glomerulus, it has connections with the Golgi cells here, and then the Golgi cells have connections with the parallel fibers of the granule cells. The parallel fibers of the granule cells in turn synapses with different cells, with the Purkinje cells, with the basket cells as well. So we move to the white matter of the cerebellum. After seeing the cells in the cortex, in the gray matter, we have seen the cells, we have seen the connections, we have seen the inputs, we have seen the outputs, we have seen the feed-forward inhibition within the neurons in the cerebellar cortex and the function of that feed-forward inhibition. So we move to the white matter of the cerebellum. So the white matter of the cerebellum is formed by afferent and efferent nerve fibers. So we have the association fibers, which connect different regions of the same cerebellar hemispheres. We also have commissural fibers, which connect areas of both halves of the cerebellar cortex. Then we also have the projection fibers, which is very important when it comes to afferent and efferent fibers entering and exiting the cerebellar cortex. So that is the content of the white matter, the inner white matter of the cerebellum. That is what forms the inner white matter of the cerebellum. So we have projection fibers, since we did not have a full explanation of the projection fibers in this slide. So I move them to the next slide. So projection fibers are the afferent and the efferent, that's the input and the output nerve fibers. They are made up of afferent and efferent nerve fibers, which connect the cerebellum with other parts of the central nervous system. That is the projection fibers. That's what makes up the projection fibers. So the projection fibers of the cerebellum are arranged in 3 bundles. Remember, they contain efferent and afferent fibers that connect the cerebellum to other parts of the central nervous system. So we have superior cerebellar peduncles, middle cerebellar peduncles, and inferior cerebellar peduncles. Those are the projection fibers. That is how the projection fibers are arranged: superiorly, middle, and inferior cerebellar peduncles. So the middle and inferior cerebellar peduncles carry afferent fibers into the cerebellum. It carries sensory fibers into the cerebellum. So that connection, that afferent connections into the cerebellum is made up of the mossy and climbing fibers. Remember, they are the two main inputs into the cerebellum. So they receive and bring proprioceptive and motor information from the motor cortex, from the vestibular nuclei, from the ready nucleus, and from the proprioceptors. That is the function of the mossy and the climbing fibers. So the superior cerebellar peduncle contains predominantly efferent fibers. That means it also contains afferent fibers. So it connects the cerebellum to the midbrain, that is the superior cerebellar peduncles, and is the main output from the cerebellum. So it has mainly efferent fibers that carries output from the cerebellum. So you can see the superior cerebellar peduncle here. You can see the efferent fibers moving out from the superior cerebellar peduncle. So you can see fibers from the deep cerebellar nuclei, the fastigial interposed nuclei, and the dentate nuclei, giving off efferent fibers that passes to different parts of the central nervous system, to the thalamus, to the radionucleus. So that is why you have the names here. Cerebello-rubral fibers. We have fibers to the the thalamus from the dentate nucleus, and we also have fibers to the vestibular nucleus, vestibular nuclei, yes. So we have vestibular fibers. You can see the vestibular nuclei here, moving out from the fastigial nucleus of the cerebellum. Fastigial nucleus is one of the deep cerebellar nuclei. So we have afferent fibers. Remember, the superior cerebellar peduncle also contains afferent fibers as well as efferent fibers, but the efferent fibers are predominant. So we have afferent fibers like spinocerebellar, anterior spinocerebellar fibers, rubrocerebellar fibers from the tectocerebellar tract, which carries auditory, visual informations as well. So these are afferent fibers from different parts of the central nervous system now to the cerebellum. So we have the middle cerebellar peduncle, the middle cerebellar peduncle. So what are the functions of this middle cerebellar peduncle? Remember, the peduncle connects the cerebellum to the pons. That's for the middle cerebellar peduncle, and is the main input to the cerebellum. So you can see the main inputs coming in from the cortex, from the cerebellar cortex. So we have the corticopontine fibers. So from the fibers from the cerebral cortex, synapsing in the pons, and from the pons, moving into the cerebellum. So it is composed mainly of afferent fibers known as the corticopontocerebellar fibers, bringing motor information from the cerebral cortex. So the inferior cerebellar peduncle connects the cerebellum. You can see the inferior cerebellar peduncle here on the diagram on the left side of your screen. It connects the cerebellum to the medulla. So it has efferent fibers and also afferent fibers. But in this diagram, we are showing here afferent fibers. Okay, we have fibers coming in, mossy fibers, bringing in proprioceptive information from the spinocerebellar tract. Then we also have climbing fibers coming in from the inferior olivary nucleus. So that is why you have the afferent fibers being spinocerebellar, humiocerebellar, and even reticulocerebellar tracts. So we also have fibers coming in from vestibular cerebellar tract and olivocerebellar tract. That's from the inferior olivary nucleus. Meanwhile, the efferent fibers are the cerebellar olivary fibers, fibers from the cerebellum to the olivary nucleus, and from the cerebellum to the reticular system. Cerebello-reticular fibers. So we also have projection fibers. Now, we're still in projection fibers. So we also have a summary of the projection fibers. We have mossy and climbing fibers, which emit collaterals to the deep nuclei, as you can see here. The mossy fibers coming in and then going into the cerebellar cortex to make connections with the deep cerebellar nuclei. So before they pass in to make connections with other cerebellar neurons. So they first of all send collaterals to the deep cerebellar nuclei, like the mossy and climbing fibers will emit collaterals to the deep cerebellar nuclei and then pass to the cerebellar cortex to synapse with the cerebellar cells, the Purkinje cells, the basket cells, and so on. So for the cerebellar nuclei, we have the deep cerebellar nuclei, which are masses of gray matter in the white matter. Remember, the inner part of the cerebellum is the white matter. So it contains some masses of gray matter which make up the cerebellar nuclei. So we have the dentate, the globus, the emboliform, and the fastigial nuclei. The globus and the emboliform nuclei are always known as, when they are lumped together, they can be known as the interpositus nucleus. So a brief explanation about the cerebellar inputs. So we have inputs coming in from the spinocerebellar tract, from the proprioceptors via the inferior cerebellar peduncle to the cerebellar cortex. So we have fibers coming in there as mossy and climbing fibers, bringing sensory information from the spinal cord via the spinocerebellar tracts. Remember, we have the two types of cerebellar tracts. We have the dorsal spinocerebellar tract. We also have the anterior or ventral spinocerebellar tract. So this cerebellar tract, these ascending fibers, terminates at the cerebellum. They enter the cerebellum and they convey information about the position and the movement of the opposite or other parts of the body. They bring information from the proprioceptors. So they enter via the inferior olivary nuclei. So from there, information regarding proprioception are transmitted to the cerebellum. So we also have other inputs to the cerebellum. We have from the cerebral cortex via the corticocerebellar pathway, as we have seen, corticopontocerebellar pathway. So we have the pathway from the motor cortex, excuse me, from the motor cortex to the pons, and from the pons to the cerebellum via the mossy fibers. So we have also from the motor cortex to the inferior olives. So if you can look at the diagram here, the motor cortex is located superiorly. So we have fibers from the corticopontocerebellar tracts, from the cortex to the pons and then to the cerebellum. So we also have motor information coming from the motor cortex and sensory cortex to the olives, the inferior olivary nucleus, and then extending to the cerebellum. So those pathways have motor information coming into the cerebellum via the mossy fibers and the climbing fibers. So the olivocerebellar tract carries climbing fibers, and the pontocerebellar fibers are known as the mossy fibers. They bring motor commands from the cerebral cortex to the cerebellum. Remember, the cerebellum is also involved in planning of voluntary movements, just like the cortex, the cerebral cortex. So fibers from the corticocerebellar circuit receive signals from the motor area of the cerebral cortex, like I said, and then conveys this signal to the corticocerebellum. Remember the parts of the cerebellum. We have the corticocerebellum or the cerebrocerebellum, the cerebral hemispheres. That is the lateral cerebellum. So information coming from the cerebral cortex terminates at the corticocerebellum. So the corticocerebellum, like I said, since the cerebral cortex and the cerebellum are concerned in planning voluntary movements. So the corticocerebellum receiving inputs from the cerebral cortex is concerned with integration and regulation of well-coordinated muscular activities. So the motor signals from the cortex to the spinal cord are also sent to the cerebellum via the collateral fibers. So we also have information that are coming down from the cortex being sent to the cerebral hemispheres or the cerebrocerebellum. So as motor information are descending by the descending tracts to the spinal cord, they also branch, or collaterals are also given off to the cerebrocerebellum. To compare, it helps to coordinate what information is being sent down to the spinal cord to carry out voluntary movement of muscles. So it will compare, the cerebellum is there to check and compare the type of movement that will be executed. So as the movement descends through the spinal the corticospinal tract via the spinal cord, there are also collaterals that are sent to the corticocerebellum so that the cerebellum can confirm and also have a copy of the motor instructions for voluntary movement, that is, as it being sent down to the spinal cord. So that takes us to the next slide here. So the cerebellum also receives information about the actual performance from the proprioceptors via the spinal cerebellar tract. We have said that before. So you can see tracts coming in, sensory tracts coming in from the proprioceptors through the spinal cerebellar tract to the inferior olivary nucleus, and then moving as climbing fibers or olivocerebellar inputs to the cerebrocerebellum or the spinal cerebellum, sorry. They move in via and terminate at the spinal cerebellum. So by this way, the cerebellum compares instructions with performance. It compares what is going on, the feedback that is coming in from the proprioceptors with the performance that was being sent down by the motor cortex. It compares instructions and performance, and then it corrects any errors. If there is any error, that is where the cerebellum will correct the error in movement. So we also have input from the vestibular apparatus, which terminates in the vestibulocerebellum. That is the flocculonodular lobe of the cerebellum, which is responsible for receiving vestibular impulses. So it receives impulses from the vestibular nerve, remember, from the spatial sense, in the middle, in the inner ear, the vestibular apparatus, via the vestibular nuclei. The vestibular nuclei located in the medulla. So it also receives eye movements from the eyes, eye movement impulses from the eyes via the points and olives would terminate at the flocculonodular lobe. So those impulses can also be communicated with the premotor area for eye movement. Then it also receives impulses. It also shares impulses, that same eye movement impulses via the superior colliculi. It still goes into the inferior olives and then down to the cerebellar part of the flocculonodular lobe, which is also known as the vestibular apparatus. Excuse me. So the importance of this connection of the cerebellum with the vestibular apparatus and with the superior colliculus for eye movement and even the occipital eye area is the importance is for maintenance of equilibrium and balance. So the cerebellum also plays a role in maintaining equilibrium and balance as a result of receiving afferents from the vestibular nerve, from the vestibular nucleus in the pons and medulla, and also receives signals from the eyes and the frontal or the premotor area responsible for eye movements. So we also have cerebellar outputs. Cerebellar outputs. Remember, the Purkinje fibers are the main output from the cerebellum. So by so doing, the Purkinje fibers has connections with the deep cerebellar nucleus before the outputs are finally exited from the cerebellar cortex. So as you can see in this diagram, you can see cerebellar afferents going in, and you can see efferents output from the cerebellar cortex via the Purkinje cells. So the Purkinje cells make synapses in the deep cerebellar nuclei before the information is now distributed either to the cerebral cortex or to the midbrain or to the reticular formation or to the spinal cord. So that is the outputs of the cerebellum. That is how it's the flow is. So in the deep cerebellar nucleus, we have the dentate nucleus, the interpositus nucleus, and the fastigial nucleus being parts, being the deep cerebellar nuclei. So from the deep cerebellar nuclei, information is now channeled out of the cerebellum to the cortex, to the midbrain, through the radial nucleus to the reticular formation. From there, they will descend. In these structures, they will now descend as the extrapyramidal tracts. From here, the spinal cord, they will also descend as descending tracts as well. So let's see. We also have output from the vermis of the cerebellum. Remember the structure of the cerebellum known as the vermis. So output from the cortex of the vermis passes through the fastigial nucleus. All these are the deep cerebellar nucleus. So from the vermis, we have output passing through the deep cerebellar nuclei, and from there to the reticular formation. Okay, from the deep cerebellar nuclei to the reticular formation. So they form the reticulospinal tract. They can descend at the reticulospinal tract. Okay, they can also terminate at the reticular formation in the pons. The reticular formation from here in the fastigial nucleus or the deep cerebellar nucleus, they extend to the reticular formation, the pons and the medulla, and then descend as the reticulospinal tract. Remember the extrapyramidal system and the reticulospinal tract functions. So from there, the lateral vestibular nuclei, it also makes connections with the lateral vestibular nuclei, which can descend as the vestibulospinal tract. Then from the intermediate zone close to the spinal cerebellum, there is output from the intermediate zone to the nucleus interpositus. Remember, the globus and emboliform nucleus lumped together are known as the interpositus nucleus. So from the interpositus nucleus, the impulses move to the thalamus, to the basal ganglia, and then to the motor cortex. From the thalamus, the basal ganglia, and to the motor cortex. They can also, that pathway can also send collaterals to the radial nucleus, and then descend as the cerebrospinal tract. And from the motor cortex, it can descend as the corticospinal tract. That shows you that the cerebellum also plays a role in coordinating muscular activities, in executing movement. So this pathway is important in coordination of reciprocal contractions. Output from the intermediate lobe of the cerebellum is responsible for coordination of contractions, reciprocal contractions of agonist and antagonist muscles. We can see such reciprocal contractions in ballistic movement. We can see such coordination in ballistic movement. For example, in punching, if someone wants to punch another person, that's a type of ballistic movement. So that type of movement is controlled by the output from the intermediate lobe. So either the radionucleus or the basal ganglia via the thalamus to the motor cortex and then descends as the corticospinal tract, or collaterals from the red nucleus descends as the cerebrospinal tract. So that tract controls ballistic movements like punching and throwing a strike. So in that type of movement, we have agonist and antagonist muscles contracting reciprocally for you to be able to make such sudden movement. So still on the output from the cerebellum, we have output from the cerebellum, from the lateral zone of the cortical cerebellum. We also have outputs from there, as well as inputs. So since we are concerned with the output, we see the flow of the output from the cerebellum. From there, cortical cerebellum to the dentate nucleus. Remember, the dentate nucleus is also part of the deep cerebellar nuclei. So from there to the dentate nucleus, from the dentate nucleus, there's a connection to the radionucleus. So from the dentate nucleus via the superior cerebellar peduncle to the radionucleus, or radionucleus, yes, and then to the spinal cord. So from the radionucleus, the tract can descend as an extrapyramidal tract, known as the cerebrospinal tract. Remember, it took its origin from the lateral cerebellar hemispheres, from the cortical cerebellum. So that is how contributions to the cerebrospinal tract occurs. So from the red nucleus, we can move to the ventrolateral part of the thalamus. From the thalamus, the next order neurons carries projection fibers to the cortex, to the cerebral cortex. And from the cerebral cortex, motor fibers can descend as the corticospinal tract. So that pathway is known as the dentato-thalamic pathway. Dentato-thalamic pathway. That is the pathway that contributes fibers to the corticospinal tract and also contributes fibers to the cerebrospinal tract. So we have dentato-thalamic cortical tract. So that is what happens in the output from the cerebellum to the motor cortex and to the radionucleus. So the output from the cerebellum provides information. As that output moves from the cerebellum to the nucleus, deep cerebellar nuclei, to the radionucleus, to the thalamus, and up to the cerebral cortex, the role of that pathway is to provide information for coordination of voluntary movements initiated by the motor cortex. Provides information about the voluntary movement that was already initiated by the motor cortex. So it also provides information used to help the cerebellum in planning. Remember, the cerebellum plays a role in planning of movement with the cerebral cortex. So that pathway is there to help. The cerebellar pathway is there to help in planning the movements that have been initiated by the cerebral cortex. So we also have other functions of the cerebellum. Control of body balance via the vestibular cerebellum, like we had seen before. Impulses coming in from the vestibular nuclei to the vestibular cerebellum. The role or the importance of that pathway is to control body balance and the equilibrium. So the cerebellum influences the spinal motor neurons to keep the body posture upright through the vestibular and reticulospinal tracts. So all those efferents we are seeing coming out from the cerebellum via the cerebrospinal tract, via the vestibulospinal tract, and down, descending down as extrapyramidal tracts are there to influence the spinal motor neurons to keep the body posture upright and also regulate the position of the eyes in relation to movement. So nucleus fastigial is the effector nucleus in that pathway. So we also have the spinal cerebellum, which plays a role in muscle tone control and then stretch reflex. So the cerebellum influences muscle tone and stretch reflex through the cerebello-olivospinal tracts. So fibers coming out from the cerebellum to the inferior olivary tracts and also to the reticulospinal tracts goes down to the gamma motor neurons. They help to influence the muscle tone. They help to control the muscle tone. So the gamma motor neurons can reflexly modify the activity of the alpha motor neurons via the cerebello-olivospinal tracts and via the reticulospinal tracts. So once the gamma motor neurons receive stimulation from the cerebellum via the previously mentioned tracts, they will help to modify the action of the alpha motor neurons, and thus they will be able to regulate the muscle tone. So the control of muscle tone and stretch reflex still through the spinal cerebellum because of that connection between the alpha and gamma motor neurons as a result of influence from the cerebellum. The cerebellum now forms an important site of linkage between the two systems, the alpha and the gamma systems, which are responsible for the muscle tone and postural reflexes. So the nucleus interpositus, being the globus and emboliform nucleus, is the effector nuclei. So control of body movement being one of the functions of the cerebellum via the cerebrocerebellum. Now, the cerebrocerebellum, like we have seen already, receiving information from the cerebral cortex. So we see the function of this part of the cerebellum known as the cerebrocerebellum. What is its functions? It is concerned with smooth performance of highly skilled voluntary movement. And it also plays a role in the planning of movements, evaluating sensory information for action, and also plays a role in cognition and speech. So the dentate nucleus is the effector nucleus for the pathway from the cerebrocerebellum. So in this diagram, you can see the flow of impulses into and out of the cerebellum and the motor cortex. So you can see the dentate nucleus here being the effector nucleus here. You can see it. So you can see the pathway, the dentato-rubrothalamic cortical pathway from the dentate nucleus to the red nucleus, and then you can see the cerebrospinal tract to the thalamus, and from thalamus to the frontal cortex. So you can also see the feedback, the afferent coming from the cerebral cortex. This pathway is involved in the control of voluntary movement, the regulation, the planning, the execution of voluntary movement. So you can see the connection between the cerebral cortex to the cerebellum. So this pathway also is known as the cerebropontocerebellar tract. Okay, the cerebroponto, this is the pons, pontocerebellar tract. So the functions of this pathway, this connection between the cerebellum and the cerebral cortex has been explained in the previous slides. So those are the functions of that pathway illustrated in this slide here. So we move away from that. We have seen the functions of the different parts of the cerebellum, and we have seen the connections of those different parts of the cerebellum, the inputs they are receiving and the outputs they are sending out to different structures of the central nervous system, including the motor cortex, and also the functions of those pathways. So we move to how the cerebellum controls voluntary movements. It controls voluntary movements via four mechanisms: comparator function, damping function, in timing and programming movement, and it also controls rapid alternating movements. So in comparator function, the motor impulses from the motor cortex via the corticospinal tract to the lower motor neurons are there for commanding movements of exploratory nature. So motor impulses from the motor cortex via the corticospinal tracts to the lower motor neurons are there for commanding movements of exploratory nature. And they are also sent to the cerebellum. Remember, there's also a connection between the motor cortex of the cerebral hemispheres and the cerebellum. So why is that information being sent to the cerebellum? I said before, they are sent to the cerebellum about the intended plan of movement. What plan of movement does the cerebral cortex plan to execute? So in the process of the cerebellum, the cerebral cortex trying to execute a movement, it will send a copy. It will send a copy of that movement to the cerebral cortex. So that is the comparator function. So now the cerebellum will play a role in comparing the movement that is about to be executed. As at the same time, the cerebellum will also receive feedback from the proprioceptive endings of muscles, tendons, and joints about the actual movement that is being carried out. So as it is comparing the feedback from the muscles, tendons, and joints, it's also receiving a plan from the motor cortex, a plan of the movement. So as a result of that, the cerebellum will compare what is going down in the skeletal muscles and what the cerebral cortex is sending to the cerebellum. So in this instance, the cerebellum may comment on every motor command via comparator function. So by this comparator function, the cerebellum also helps to provide smooth coordinated movements of the limbs for performance of purposeful patterned movement. So we move to another mechanism by which the cerebellum helps to control voluntary movement. We have damping functions of the cerebellum. The cerebellum can prevent overs. It prevents one from making movements that are over the targets. So all movements are pendular with tendencies to overshoot. But the cortical cerebellum sends impulses to the cerebral cortex to discharge appropriate signals. So as the cortical cerebellum is sending impulses to the cerebral cortex, it helps the cerebral cortex to discharge appropriate signals to the muscles, thereby preventing any overs and overshooting of a muscular activity. It will prevent an exaggerated muscular activity through that connection. Timing of movement and programming the movement is also a mechanism used by the cerebellum to control voluntary movement, timing of movement. So the communication between the cerebellum and the cerebral cortex provides a planned sequential movement. A planned sequential movement involves transmitting one movement to another. For example, taking steps, walking, one step after the other. So the cerebellum and the cerebral cortex have communicated with each other so that once you take one step, the next thing you do is to take another step. That's a planned sequential movement. And it also provides timing function, predicting events in movement patterns. It provides timing function and also plans the sequential movement. So learning processes in some cases might be able to overcome the planned sequence movement. For example, there are some movements one can take without even thinking about it. It's involuntary. So learning processes can help to overcome the steps of planning sequential movement. Then the cerebellum also plays a role in contributing to rapid alternating movement. Rapid alternating movements like dancing, pronation, and supination movements are also controlled by the cerebellum. So we go to the cerebellar diseases. And I wish you would read up the cerebellar diseases. You have seen the functions of the cerebellum, the connections with other parts of the components of the central nervous system, and how the cerebellum is involved in planning and execution of voluntary movements, and the mechanisms that the cerebellum uses to check the execution of voluntary movements. So the cerebellar diseases, damage to the cerebellum, will throw more light on the functions of the cerebellum. But the main characteristics of cerebellar diseases includes mainly ataxia and intention tremors. Ataxia is very, very pronounced in cerebellar diseases. So there are motor damages that are associated with damage to the cerebellum. The damage varies. The abnormalities damage depending on the area or the part of the cerebellum that is damaged, but the characteristic abnormalities that are exhibited in cerebellar diseases include hypotonia, ataxia, and intention tremors. So I would like you to read up the applied physiology of cerebellar diseases, the presentations, and the manifestations of cerebellar diseases. So I thank you very much for listening, and I hope you get back to me the correspondence if you have any questions, and if you have any concerns about any area that is not clearly understood in this topic. So you can contact me on my phone number there, or you can write me an email on my email address there. So thank you very much for your time, and thank you for listening, and stay safe.